Today we'd like to introduce you to the basics of cell anatomy as it pertains to the functioning of the lysosome. In order to understand how lysosomal disorders create many problems for the cell, a brief review of cell anatomy may prove helpful. The cell is surrounded by a semi-permeable membrane composed of lipids and proteins. Within the cell is the cytoplasm that contains thousands of amorphous proteins as well as many cell structures called organelles, which are pictured here. Let's take a closer look at these. Organelles include the mitochondria, which produce energy through the process of oxidation, the endoplasmic reticulum, the Golgi apparatus, and lysosomes that degrade proteins targeted for recycling. Organelles can be associated with or tethered by a network of microtubules extending through the cytoplasm, originating from the central area appropriately called the centrosome. The cell nucleus is surrounded by a membrane that contains gaps called nuclear pores. These gaps allow molecules to exit and enter the nuclear area. Within the nuclear membrane are the DNA and a prominent structure called the nucleolus. The nucleolus helps process RNA messages produced from the DNA to get them ready for exit from the nucleus. Many proteins, such as the hydrolytic enzymes, will eventually be incorporated into the mature lysosome, noted here. These proteins ultimately derive from information sent from the cell nucleus that is encoded into the messenger RNAs. These messengers leave the nucleus through the nuclear gaps to interact with ribosomes. Ribosomes produce proteins that are entered into the endoplasmic reticulum. The proteins are then processed and leave the endoplasmic reticulum to enter the Golgi apparatus. The Golgi then performs additional important protein modifications in order to make them fully mature. Proteins then leave the Golgi to be transported into the lysosome.